Good afternoon, everyone. This is Joseph Emil Rubico, Technical and Marketing Manager for Hipra Philippines, and I am here to introduce to you one of our distinguished speakers this afternoon. She is a Doctor of Biochemistry and Molecular Biology of the Universidad Autónoma de Madrid, with a PhD in Molecular Biology from the same university. Her research on virology involves mechanisms of viral cell interactions and the development of antiviral tools such as vaccines. Besides being the coordinator of IF and ASF, a project of the International Coordination of Research on Infectious Diseases, she is also the leader of the ASF consortiums in Europe and China, and the leader of several research projects worldwide. And without further ado, to talk about ASF vaccine updates and why do current vaccine fail, let's all welcome Dr. Yolanda Revilla.
Good afternoon, ladies and gentlemen. It is my pleasure to be here with all of you. Thank you for coming. I would like to thank the organizers for inviting me, and also I am very pleased of honor Dr. Marcial Ramos, that is kind of great precursor of the care and research of swine. So, I'm going to talk to you all about the African swine fever, and I am going to talk about both the virus and also I am trying to describe why do current vaccines fail. Well, African swine fever is a complex DNA virus against which no fully efficient vaccine exists so far. The disease emerged from Africa in 2007 and goes to Armenia in a ship, probably in contaminated food or rests. So when arrived in Georgia, it rapidly distribute or spread about around the neighbor countries, especially Russia, and then from here to Europe, Oriental Europe, and especially it is still endemic in Poland, Bulgaria, Romania, Latvia, Estonia, Lithuania, and so many countries in Europe. Although regionalization measures from the Commission have been very useful to control the virus, the virus is able to infect wild boar, and wild boars, as you know, are a species of swine that rapidly moves into the territory, so they rapidly spread the virus. Also, it is important to know that there is a vector, and is a soft tick between the domestic pigs and wild boar, and these ticks is able to replicate the virus in it. The insect is not infected by, in terms of they can maintain the virus replicating, and is for years, and is very typical to transmit by biting the animals the disease. So the current scenario about African swine fever virus spread throughout the world involves several countries, as you see here, apart from the countries from Oriental Europe, Russia, that I have told you before. Very lately, the virus in Europe has also emerged in Germany, this summer in Italy. Importantly, the virus in 2018 arrives to China. From there, it moves to East Asia involving several countries there, like Philippines, Korea, Vietnam, Cambodia, etc. And also very recently, the virus has jumped to the Caribbean part, infecting or producing an outbreak in Dominican Republic and Haiti. So this is a very important issue because this is the closest movement of the virus to America. Um, Regarding the current situation in Asia of ASF, as you probably know, the virus, apart from China, which is completely, infecting by the virus all the country, also, unfortunately, Korea and South Korea, and also Philippines, Cambodia, Vietnam, and all these neighboring countries around China, Taiwan, are suffering this disease. That is a very serious disease for um, the swine, giving very important economical problems to the sector as it's very difficult to keep the virus into the control measures. Indeed, in Europe, as I said, regionalization processes have been planned from 10 years ago. However, the virus emerged suddenly in Italy, or like it happened this summer, and I mean it's unexpectedly emerging everywhere, probably due to its ability of infecting wild boar. As you can see here, for instance, in Korea, I mean the current situation is very worrying because the virus is completely taking the country now. It's also over here at this moment, and well, this is a really worrying situation for all Asia. Like this virus, although is not able, fortunately, to infect humans, I mean, it's a virus that causes a really serious disease with different amount of severity and virulence. More virulent strains kill the pig in around 10 days. I mean, as I said, in ticks, the virus can replicate, but doesn't kill the insect, so it's also a worrying factor. And wild boar used to be more resistant to the virus. In this slide, you can see how epidemiological studies have established the way in which the virus emerged from China, and it's so easy for the virus to emerge in food, in waste, and in contaminated materials, which are very difficult to be controlled, unfortunately. So I'm going to tell a little bit, just a little bit, of the complexity of the virus, which contains a double-stranded DNA, very large DNA of 170 kilobase pairs. This DNA encodes for more than 170 proteins that interact with the host, with infected host, controlling several mechanisms and evading most of the mechanism of the immune response of the host. I mean, why are ASF vaccines not available so far? Having in mind the complexity of the virus, you can see here how the virus emerged from the infected cell in a picture of electron microscopy. So this virus is so complex that encodes for so many factors and proteins, and these proteins are not able to induce antibodies in terms of neutralizing the infection. So if you compare with COVID-19, against which fortunately in less than 2 years efficient vaccines have been developed, the difference with this kind of so complex viruses is that whereas in COVID-19 you can find a spike protein, is the ligand of the virus, is the protein that the virus used enter enter the cell. In the case of African swine fever, there is only one ligand, and also the proteins that are candidates to be the ligand do not induce neutralizing antibodies. In the case of COVID-19, neutralized antibodies against spike fortunately neutralize the infection. Thus, the vaccination can perfectly control the virus only by putting RNA from codifying for spike, or only with recombinant products of the spike protein. There is no spike protein in African swine fever virus as far as we know. And this so complex structure containing a DNA into a nucleoid in here that is surrounded by a core cell, an inner membrane, then the capsid that encodes very important proteins of the virus that you can see in this graph. But not only that, and also this outer envelope, the virus gets it from the infected cell. So it's kind of a coverage to be evading the recognizing by the immune response of the host. So in this situation, in our lab from many years, 20 years ago, we were asking how we can do a vaccine against this virus. It is important to know before that as I told you, there are different levels of virulence in the strains that are circulating, and the virus, as all the rest of the virus, is able to get more and more attenuated. So some of those attenuated strains, for instance, these naturally attenuated strains that appears in Portugal in 68, when we here in Spain and Portugal, we had the disease. I will tell you later about this attenuated strain. We have used it to make as early as 2016 and 2017 several vaccination studies by using these attenuated strains. Also, we used these attenuated strains to make recombinant viruses that lack of different genes that we were characterizing by that time, and we do in this. Indeed, we did studies about the protection that this attenuated and recombinant virus can induce. So from there to now, the question is: safe ASF vaccines Are they feasible? Well, if you look around, we can see that apparently in Vietnam, by using a vaccine that has been done by our colleagues in Plan Island, these guys have done a recombinant vaccine that apparently has been produced by Nedbeco and with this company, they have developed a vaccine that apparently protects against the virulent strains. However, side effects or viremia, I mean shedding from the vaccine, that this is not really a thing that is good for a woman at the vaccine, as you can spread too much this kind of material, and this can have some problems in terms of diffusion of uncontrolled vaccines from animals, from vaccinated animals to other animals. But also, they have found that some of the animals that have been vaccinated apparently died. So, though it's true that you cannot tell that the vaccine is the cause of the death of these animals, it's also true that vaccine with live attenuated virus, although is the more realistic method to produce an African swine fever vaccine, I mean, can have some problems on safety. This is apparently also what happens with the approach that currently is being developing in Revilla farm here in Philippines, although no much information I know about this vaccine, so maybe you know more than I about this product, but under my opinion, I mean this vaccine is still being tried. So the question is, what are we doing in my lab about vaccine development? As I told you, we have been involved in vaccine development for more than 20 years, and we have produced and tested subunit vaccines too, because this could be the best solution if working, but unfortunately don't. So we have tested some combination of ASF proteins and DNA. We have been tested inactivated vaccines by inactivation of the virus with radiation, no success at all. So we are also studying vaccines based on live attenuated strains, or based on a deletion of a specific genes that we have studied are involved in virulence, to construct new prototypes from the circulating strains lacking of these specific genes. Also, we are all currently looking for non-replicative vaccines, what we call next generation vaccines, but indeed this is just in the pipeline. I mean, it's just only wishful thinking. So what I can tell you about what have we done. We have used inactivated virus, as I told you, and we did subunits, DNA-based vaccines, and all attempts have failed to confer full protection against lethal viral challenge, as you will see in the next slide. So we are now working with live attenuated vaccines that, as I will tell you, result in a quite interesting and promising results. So one of the most important trial that we did combine cocktails of DNAs, specific DNAs from genes that we have characterized it being important or very P72 for instance is the most abundant gene and protein, I mean gene in terms of expression of the virus, and we did a series, a set of cocktails, and we vaccinate animals, but unfortunately 100% die after challenge with the circulating Armenia strain. So though we have been working very hard with this kind of preparations, they have resulted failed, all of them. We move then to natural attenuated strains. Natural attenuated strain like non-hemosorbing Portugal 68 coming from the 80s. I mean, when but that time Spain and Portugal we were endemic, and it took us 30 years to be free of the disease because no vaccines by that time, of course, and only killing with programs of killing pigs, we can get free from the disease after 30 years. So we have tried what happens with these non-hemosorbing strains of the virus, naturally attenuated strains. There are new attenuated strains also from Latvia and other countries because, as any other virus, African swine fever virus, is getting attenuated, whereas the disease is progressing, as every virus normally in the world. No, they prefer to be attenuated. They are not interested in killing the animals, but during this attenuation period, several grades of the disease are present in these animals, so you cannot use this kind of genetic background, this kind of parental virus, just to immunize the pigs, because you will get this kind of undesirable side effects. So with this in mind, we have tried a different strategy that involve the manipulation of the genome of the virulent strains, and especially the circulating strain, both strains like Armenia, O7, or Korean Paleo strain, that we are manipulating by using a technique that we have developed that is called CRISPR-Cas9. By using this technique, you can kind of editing the genome of the virus in such a way that you can substitute a specific gene for a marker. By doing so, we have got green viruses that contains GFP and other markers, and however lacks from the gene we have eliminated. So why we have eliminated these genes? Because we have been studying for several years, we have spent several years in discriminate what is virulence, what does virulence means, and what does attenuation means. So as you know, there are strains that are cause acute infection, strains that are really virulent, like Armenia 07, the Chinese strain, the Korean Paleo strains, and other circulating strains that just kill the animal in around 10 days. Then other kind of subacute infection with variable percentage of mortality can emerge, and at the end of the years you can see persistent infection that show clinical lesions but are attenuated and do not kill the animal. This is represented by non-absorbing P68, for instance. So what are the genes? What difference can you get in this between these virulent strains and these attenuated strains? What are the difference in terms of the genes that compose the genome of the virus? So we have studied this in deeply, and we have described what are these genes in terms that we have compared genomes coming from virulent versus genomes coming from attenuated strains. So we have identified why the virus does in the nature. What are the genomes areas that the virus reorganize or rearrange to get attenuated? So we have focused on these genes, and also we have focused in genes that we know that are involved in cellular immune control. That means that the virus, African swine fever virus, can absolutely control, for instance, the gas-steam pathway or JAK-STAT pathway that are critical in the control of innate immune response, especially in the control of interferon beta production. Interferon type 1, you know, that is one of the first cytokines that start the innate immune response and produce the scenario for the secondary specific response start. So in this regard, we have identified several genes. One of those is the CD2V, that is one gene which is only expressed in virulent strains. And we have described that localized around the viral factory. You can see in this picture an interesting pattern in which you can see here one infected cell. This is the nucleus of the cell, and this also in blue is just the factory, the viral factory that contains the viral DNA, also in blue like the cell DNA. And you can see here how we stain this factory with an antibody staining the P72, the most abundant protein of the viral factory. And this factory is importantly surrounded by this gene CD2V. So we hypothesize that CD2V is an important gene for the virus. So what happens if what could happen if we delete this gene? Not only that, but also we have been studied for many time ago that there are other genes, for instance, 8238L, that is a viral gene that is able to control the immune response. And we have described the mechanism by which this gene controls the immune response. So it's interesting to mention that the virus encodes this gene that is an inhibitor of a nuclear factor is NF-kappa B, which is a transcription factor for so many pro-inflammatory genes and factors. So this gene by itself, A2CHL, is able to control TNF-alpha expression in the host, the nitric oxide inducible expression, the COX-2 activated pro-inflammatory factor. So this gene is indeed controlling cytokines that are critical for the defense against this virus. So we have already two genes that we consider very important, CD2 And 8238 cell that we lately we have also described that it is able to modulate the interferon production, interferon type 1 production. And we have also studied the model. I don't want to be very much on this results because they are too molecular, maybe, but just to tell you that interacts, we have clearly seen the mechanism that use the interaction with one of the IKK. There are kinases, really important to, to the pathway in which the interferon production occurs. So we have focused these two genes, CD2 and 8238L, that are viral genes important in the control of the interaction of the cell and in the interaction with the immune response in the host. And we have deleted both of them from the genome of Armenia. Okay, Armenia 07, that is the virulent strain circulating in a big part of the world. Is the circulating strain also in South Asia, East Asia, and in Philippines or in Korea, and nowadays. So by using our CRISPR-Cas9 technique, we have been able to modulate, to delete these two genes. And you can see here, this is the demonstration that this virus lacks both of CD2B and A238L gene, okay? Whereas the markers GFP and Cherry substitute these two genes. As you can see in the wild type control, on the contrary, the expression of CD2B and A238L is patent, and the absence of the markers, of course. So this is a prototype that we have generated in my lab, and with this prototype we have vaccinated as you want, pigs, and we have shown that after the challenge, first of all, we have demonstrated that these vaccinated animals do not are affected by this virus. So the first important thing is that this virus is already attenuated. Coming from one very virulent strain. However, this virus that we have created is an attenuated one. So, because you see here, compared with a control non-vaccinated, you can see that the animals do not die, and also that if you challenge these animals with virulent strain, Armenia in this case was the Korean Pajo strain, very virulent. The animals keep alive, comparing with the non-vaccinated that were all died. When we have studied the situation in the animals, in the vaccinated animals, in terms of antibody, we can see here in red that the animals vaccinated with our prototype is a very, very strong and rapid antibody response. You can see here that after 10 days of vaccination, antibodies growth in the animal, and you can see, comparing with other kind of candidates and comparing with non-vaccinated animals, we have a very important and very strong curve of neutralizing antibodies. Not only that, but also one of the most important concerns when a vaccine is developed, of course, a live attenuated vaccine, that is an attenuated virus, you need cells to grow that. The problem with African swine fever virus is that it's a virus that have a very strong tropism and only infects in the animal monocytes and alveolar macrophages. So other prototypes that are currently trying to be introduced in the market indeed are vaccines that need to be grown in alveolar macrophages. However, our prototype we have been studying also for many years is not something that we have from now. We have been studying this from many years ago, and we have a model to grow our vaccine. So our current cell models involve was one that is cell from ATCC, so you can just grow the virus in here, which is really important for the scaling up of the vaccine industrially. And also we have another lines like a WSL comes from wild boar. We have developed other cell lines that you can just use to grow the vaccine. This way, get over the problems that using macrophages that are just primary cells that comes from cells, live animals, live pigs. This, of course, is not very convenient because you can expect different behavior among batches, different batches, also contamination from the animals, and also at the end of the day, ethic consideration of animal welfare. So, in conclusion, regarding vaccine prototypes and regarding our vaccine, we have found significant attenuation, full protection by a mutant developed in our lab. Still, the research is focused on the genetic stability of this prototype that we have got. That is, we have found that is really stable in vitro, in passages in vitro. You can pass the viruses more than 10 passages and you still get the genome of the virus fully stable. Also, we are doing in vivo specific studies now to see the genetic stability of the virus when you pass it in animals, just to further confirm the stability. Also, we are focused on DIVA tests, and DIVA tests, as you know, are tests that involve the determination, the differentiation among vaccinated versus non-vaccinated animals. That this is very easy in our model, as we can just amplify the markers to see that the animal is vaccinated. But also, we are developing tests to control serology among naturally infected and vaccinated animals. Also, doses is research that still is ongoing. However, we know that our product protects at doses so low as 10 to the 2 intramuscularly by in each peak. This is really easy to scale, but because we normally get about 10 to the 8 in one purification, not industrial, just in my lab. So the study of doses will be very soon establishes that the doses is 10 to the 2, but we can go even lower. And also we are going to demonstrate that 10 times more, let's say 10 to the 3 for animal, is not going to be damage for the pig. Also, other parameter is to be studied is the setting of the vaccine and sentinel studies that we are already developing. Um, as far as we know, until now, the setting of the vaccine, I mean the viremia in the infected animals in blood and also in other secretion like swabs or fecal restamine is very low. You cannot find the virus in here, so this is good to is good avoiding the setting of the vaccine for safety reasons. And also, at the end of the day, we are trying to get with interested companies that are not only Hipra, of course, but also in Korea, Kersai, that has been also supporting our research until now, and I thank them. So just to finish, I let you know the composition of my group, that is composed by postdoctoral researchers and predoctoral fellows and technicians that to them I thank you very much for their hard work. Also, we keep collaborations with Dr. Garcia Sastre in Mount Sinai in New York City for the non-replicating new generation vaccines that we are developing right now. And our thanks to our industrial partners, IPRA, CareSite, and lastly, Avimes. So thank you so much for your attention, and I hope you have some questions, and I will be delighted to answer them. Thank you very much for your attention.